So hello all, I welcome you to the new video. So we have completed the amino acid series. Now we're going to start the nucleotides. I hope you can see my achievements in this section here. I have qualified AIMS PG entrance for biochemistry with All India rank 3. I have also qualified CSIR NET and other national level entrance examinations like GATE, IIT JAM, etc. etc. So today we're going to discuss about the tricks to learn, nucleotide structure and how nucleotides are being formed, what are the different kinds of nucleotides and how DNA is formed, how RNA is formed, etc. etc. So as we all know, so let's start the topic now. As we know that our genome is responsible for all the phenotypic expression that we have, the eye color that we have, the body stature that we have, the height that we have, for everything our genome is responsible for that. Our genetic material is responsible for that, the unit of our hereditary, right? So this is made up of genes, right? The genes are on the genome. These are made up of nucleotides, right? Now what are these nucleotides? Nucleotides are made up of 3 units. Okay, so it is made up of sugar, nitrogenous base, plus phosphate group or phosphoric acid. So this is the composition of nucleotide. Now there are 5 types of nucleotides on the basis of the different nitrogenous bases. Sugar phosphate backbone remains the same, but the difference comes in these nitrogenous bases. Now these nitrogenous bases are divided into 2 categories: purines and pyrimidines. These we all know, right? Now purines are further divided into 2, that is adenine and guanine. And your pyrimidines are divided into cytosine, uracil and thymine. So we will discuss the structure of these nucleotides in detail today. Now this is the nucleotide, right? This is the nucleotide. Now there is another term which is known as nucleoside. What is called? Nucleoside. Nucleoside is made up of the sugar and the base. Only the sugar and the base. It does not contain the phosphoric acid group. The nucleoside contains only sugar and the base, and your nucleotide consists of sugar, base and phosphate group. Okay, we'll discuss the names of nucleoside and nucleotide further in detail, and I'll also solve like 2-3 questions also for you at the end of this topic. Okay, now we're going to draw the structure of the purine ring. Now these purines and pyrimidines are derived from a basic ring structure which I'm going to draw for you, and those purines and pyrimidines are derived from those rings only. Okay, so today now I'm going to draw the structures of purines and pyrimidines. All right, all the concentration on the board. So purines are made up of double ring structure. Okay, purines are made up of double ring structure. Always remember this thing, that purines are made up of double ring structure, and purines contain what? Ag. Purines contain what? Ag. You have to remember this thing. Purines are A and G. That is Ag. Okay. Purine is made up of, purine is derived from this double ring structure. Now the numbering of this ring is very crucial. That numbering of this ring is very crucial. So for purine ring, the numbering is anti-clockwise. So we number from here 1, 2, 3, 4, 5, 6, now 7, 8, 9. So this is the numbering of the purine ring. Baki the carbon atoms and everything we can attach later on, right? So this is the numbering of what? Your purine ring. Now we are going to learn the numbering of the pyrimidine ring. So you can see that this is anti-clockwise. So the purine ring numbering is anti-clockwise, and it starts from here 1, 2, 3, 4, 5, 6, then continue from here 7, 8 and 9. So this is the purine ring numbering, and this is how you draw a purine ring. Double bonds we can attach, nitrogen we can attach when we draw the structure of adenine and guanine. Right? Now, as I have shown you the basic structure of purine, now I'm going to tell you how to draw the structure of adenine, adenosine. Adenine, because adenosine is your nucleoside. Adenylate is your nucleotide. Adenine is your nitrogenous base, and right now we are drawing the nitrogenous base, and it is derived from the basic purine ring which I had just drawn, right? So what are the features of the purine ring? It is double ring structure. Its numbering is in anti-clockwise fashion, okay? And there are 9 total groups that are attached to that ring, all right? So these are the basic features. It is double ring structure that you have to remember thoroughly. Okay, now I'm going to draw the structure of adenine for you. Adenine is also called 6-amino-purine. So to the purine ring, you have to attach amino group at the 6th position of the purine, alright? So we are going to draw the purine ring now. This is the purine ring. So I'll just give the numbering 1, 2, 3, 4, 5, 6, 7, 8, 9. So what did I just say? That on 6th position of purine, we have amino group attached to it. So it is 6-amino-purine. It is 6-amino-purine. So we have drawn the structure of adenine, that it is 6-amino-purine. 6-amino-purine. The double bonds and everything you have to see yourself, okay? So this is 6-amino-purine. On the 6th position of the purine ring, there is NH2 group that is attached to the purine. So we can call the adenine as 6-amino-purine. Now the next structure that we are going to draw is your guanine structure. So in the structure of guanine, you have to shift this NH2, shift this NH2, इसको खिसका दो, धक्का मार दो नीचे की तरफ, यहाँ पे इसको धक्का मार दो नीचे की तरफ, then you will form the guanine ring, right? So guanine structure. So what you will do is you will shift this NH2 on the 2nd position, and here it comes the double bond O. Okay, here it comes the double bond O. So here we have NH2 niche, and here we have come with the double bond O here. So we have adenine and guanine. So guanine, adenine is your 6-amino-purine as we have discussed already, and this is the structure of guanine. So we have done the true structures that of purine rings, that is adenine and guanine, that is this structure. Are we clear? I hope we are clear with that, right? Isko, when adenine, if we talk about adenine, then that is 6-amino-purine. On the 6th position, amino is attached. For guanine, for the structure of guanine, you have to shift this amino group नीचे, इसको नीचे खिसका दो और ऊपर यहाँ oxygen atom आ जाएगा. Okay, so we are done with both the structures of purines, that is adenine and guanine. Okay, now we are going to draw the structure of pyrimidines. Now we are going to draw the structure of pyrimidines. Pyrimidine is a single ring structure. Pyrimidine is what? It is a single ringed structure. All right. So in the pyrimidine ring, the numbering starts from below here. 1, 2, 3, 4, 5, and 6. And the numbering is in the clockwise fashion. You can see, right? 1, 2, 3, 4, 5, 6. In the pyrimidine ring, we saw that the numbering was in anti-clockwise fashion. In the purine ring, it was anti-clockwise fashion. But in the pyrimidine ring, it is clockwise fashion. Alright. Now, pyrimidines are further divided into 3 categories. 3 nitrogenous bases are there that are included in the pyrimidine category of nucleotides. So we have CUT. What were purines? They were AG and it is CUT. So purines are AG, this is CUT. Just say it 5 times, you will be able to learn it properly. AG is purine, CUT is, cut is your pyrimidine. Okay. So pyrimidine is your clockwise fashion. The pyrimidine numbering is in the clockwise fashion. Okay. Now we are going to draw now we are going to draw the structure of C, U and T. Okay. So as we know that A pairs with T and G pairs with C. In the GC structure, we have drawn na. Now I'll draw it like this only. Okay. In the GC structure, what did you do? O was here and NH2 was here, right? So you have to draw the complementary structure in case of cytosine, right? Because G is complementary to C. So it's the trick to learn how to learn the structure of these, pyrimidines and purines. So in the pyrimidine ring, the G was like this. and C pairs with G with the triple bond. So it has to be the complementary structure of G. So what will happen in the structure of C? This will come upar, this will go niche. So it will form the complementary structure with the C. So if we draw the pyrimidine ring, then we will get NH2 upar, ye oxo niche. So this is your what? C ka structure. This is your cytosine structure. Double bond you have to adjust on your own, and the nitrogenous atoms also. Okay, so we have to draw, as you know that G pairs with C with the triple hydrogen bonds. So G ka complementary you have to draw the C. If oxygen is upar, it will go niche in this structure. NH2 is niche, it will go upar in this structure in the case of cytosine. Okay, so both our structures are complementary to each other and they bond with each other with the triple hydrogen bonding, right? Now we are going to draw the structure of uracil. Sorry. Now we are going to draw the structure of uracil. So in the structure of uracil, what will happen is the structure of uracil. Uracil is formed by the deamination. Very, very important question with your exam point of view, that the deamination of cytosine yields uracil. The deamination of cytosine yields uracil. So you remove this amino group from here and put the oxygen atom. So this will form the uracil. Very, very easy to learn. So I'll repeat it for you. C was complementary to your G. So in the G, NH2 was here. So in the C, NH2 came here. In this G, oxygen atom was here. So in the structure of C, the oxygen atom came here. Now for the uracil is formed by deamination of cytosine. It is formed by deamination of cytosine. So what will it form? In the place of NH2, you will deaminate this structure, then the oxygen atom will be attached here. Alright. Now we are going to talk about the thymine. Thymine. How is thymine formed? Just to the structure of your uracil, you have to add a methyl group. Okay. Pyrimidine is single ringed only and T is your single ringed only. That's what we know. So to the structure of uracil, we have to add one methyl group. So we have drawn the structure of all the kind of nitrogenous bases. They are very, very important because in your question papers, usually the questions are asked, write the structure, draw the structure of 7-methyl guanosine. Draw the structure of ATP. So these kind of questions will be asked. So for that, you actually need to know the structure of these nitrogenous bases. So let's just revise what we did today. So for the purine ring, in the purine, we have two categories, AG. For the pyrimidine, we have three categories, CUT. So AG and CUT. Purines are double ring structure. Your pyrimidine is a single ring structure. In the purine, the numbering runs in what fashion? In anti-clockwise fashion. In the pyrimidines, it runs in the clockwise fashion. Then in the purines, we have adenine and guanine. In the pyrimidines, we have cytosine, uracil, thymine, that we know. And we have also drawn the structure of them. Adenine is what? 6-amino purine. And in the structure of adenine, you just shift the amino niche, then the oxygen atom will come upar, that will be your guanine. And you do the opposite of that, then cytosine will come. Then you deaminate cytosine, uracil will be formed. You deaminate cytosine at this point, then at this numbering, then uracil will be formed. Then you add a methyl group to your uracil structure, then your thymine will be formed. So these are the all structures of nitrogenous bases. In the next lecture, we will discuss about how to form the DNA, the sugar phosphate backbone, what are the different kinds of bonds that are involved in the formation of the DNA. Maybe you can write it in the chat sections, what are the important bonds that are involved in the formation of DNA. Okay, and that's what we will do in the next lecture. Thank you so much for watching this video. Bye-bye. Like, share and subscribe. Thank you. Bye-bye.